So thanks for the introduction. So the topic of our project is named like the graph-based spatial transcriptomics computational methods in kidney diseases. Why we are trying to do the kidney-related diseases? Because people may know that 1 of the 7 people in the world, they are encountered by the kidney diseases named like the chronic kidney disease, CKD. And up to 5 to 7 million of the patients with the kidney failure, they have to do the renal replacement therapy. And another very important kidney disease named like the acute kidney injury, AKI, can lead to the CKD and affect other organs. People know that the morphological and cellular changes occur in the renal tubulus and the surrounding interstitial niche. And the emergent spatial transcriptomics enabling the spatial anchoring of the biomedical markers that are related with the kidney injury. I think the biggest challenge in modeling kidney-related spatial transcriptomics is that the kidney has a very, very heterogeneous and mosaic-like tissue architecture. So we know for one kidney, it contains more than 1 million of the nephron and consists of the glomerulus and the tubules. So the data we used in this project mainly come from 3 data connections. So first, everybody uses is the human cell atlas, and the HAP map, which contains most of the healthy kidney tissues. And the KPMP, which is the full name is the Kidney Precision Medicine Project, which contains both the healthy and the diseased kidneys. So actually, at that time, this is the only 3 data connection we can find with the kidney spatial transcriptomics. So our project proposed to develop an unbiased, robust, and interpretable deep learning framework for spatial transcriptomics data analysis on chronic kidney diseases and acute kidney injury. So our first aim is trying to build a relation equivalent graph neural network to model this highly heterogeneous and mosaic-like kidney spatial transcriptomics. We use lots of different technology to improve the power of the graph neural network. And then build on that, we're using the multi-grained self-supervised learning to compare the healthy and the diseased kidney to illustrate the key biological mechanisms. So just like fibrosis, epithelial repair, and the immune localization. And finally, we predict how severe and how big the kidney injury is with a diffusion process. So with this project support, we developed a new methodology named like the BSP to identify spatially variable genes. And that work is highlighted by editors' focus on biotechnology and methods in the Journal of Nature Communication. And using one sample from the acute kidney injury, we identify 2 groups of the genes. So one is related with the kidney and the epithelial development. And the second one is related with the immune responses. And they are consistent with the biological knowledge. And we then connect cell states proportion, spatial variable genes, and pathway correlation, and united them together by using SPH GFT, which is just accepted in the Journal of the Nature Communication. And with the help of the cell atlas of the kidney injury published in the Journal of Nature by one of our multiple PI, so Dr. Michael Edon, we identify the injury localization showing the enrichment over two very important cell states, so APT and DPT. So this injury mapping and localization of APT and DPT with a spatial variable gene demonstrating their role in addressing kidney injury signals. So I want to emphasize, so this is a whole group efforts, so like a interdisciplinary multiple PI team. So, I myself is trained in computer science, and I worked in the department of biomedical engineering and informatics at Indiana University. I do AI methodology development. And Dr. Chima, who is just sitting there, so he's training in mathematics, and he's from the Ohio State University, and he is an expert in the biomedical informatics. And the third multiple PI is Dr. Michael Edon, who is trained as a physician, and he did a lot of the data sequencing. And he's from the School of Medicine of Indiana University. And I want to mention that he is named as the top doctor of the kidney diseases in the Indiana state since 2018. And he plays a key role in the biological and clinical support in kidney research. And I want to thank the PhD students, the postdoc, and the consortium, and the NIDDK and ODSS. Thank you. Any questions?
Thank you so much. We have time for some questions. Yes, please. Thank you.
Thank you. It was very nice work. I guess one thing I wonder is with spatial transcriptomics, the sample sizes are still quite limited in many cases.
Yeah.
And I was wondering if you've given consideration to how you can aggregate samples that may look quite different with your methods and see if the results are generalizable.
Yeah, let's go. So I think that is a great question. Yeah, so just like you mentioned, when we're doing this kind of the work, we have very limited over the data. So one of the solution we are doing is a self-supervised learning. So we can try to do some augmentation of the data and try to get good results. And we also, so from the perspective of the methodology development, we also try to use some other organs to just develop the methods and fine-tuned in the kidney.